Assalamualaikum. So today we are going to discuss about some basic concepts of microbiology, in which we'll start with the basic terminology, that is bacteria, and basically the entire structure of bacteria. What is it composed of? What are the special structures, and how different envelopes which surround the bacteria, they help in the protection of bacteria from the harsh environments. So, uh, now the topic of our today's discussion is bacterial structures. For that, first of all, we'll have a look on the structure of bacteria, and after that, we'll discuss the details. So you can see right over here, there are two types of bacteria: gram positive and gram negative bacteria. This is gram positive and this is gram negative. Now, there's some structural differences in gram positive and gram negative bacteria, and they're differentiated on the basis of stains. So, for example, we can see that this lipotecoic acid is unique to gram positive bacteria. Similarly, we can see that in the outer membrane, the outer membrane is present only in gram negative bacteria while it's absent in gram positive. So these are some of the structural differences. Now let's move on into the details of the bacterial structures. So the um, first of all we have the appendages. Uh, Flagellum, its chemical composition is that of proteins and its function is the motility and movement of bacteria. तो एक जो बैक्टीरिया है उसने जब भी एक जगह से दूसरी जगह पर ट्रैवल करना है तो वो जो स्ट्रक्चर जो उस चीज़ को हेल्प कर रहा होगा दैट इज़ फ्लैजेलम. The second structure is pilus or fimbria. It's chemically composed of glycoproteins. Uh, and what is its function? There are two types of basically pili. There's the sex pilus and there is a simple form of pilus. Pilus basically mediates the adherence of bacteria to cell surface. So adherence is the normal general function of the pilus or the fimbria. But specifically, what does the sex pilus does? What does the sex pilus do is that it helps in conjugation, mating, right? So in this figure, we can see that this is the pilus, and this is the flagellum. And flagellum and pili both are present in both types of the gram positive and gram negative bacteria. So, wait a minute. All right. I'm sorry. Acha. Now, what are the specialized structures of bacteria? They are the spores. Spores क्या होते हैं कि बैक्टीरिया जब अनफेवरेबल कंडीशंस होती हैं तो बैक्टीरिया एक ऐसा स्ट्रक्चर बना लेता है जो कि उनको उस हार्श एनवायरनमेंट से प्रोटेक्ट करता है फॉर सम टाइम बीइंग. ठीक है. इस स्पोर की फॉर्मेशन क्या होती है? डेफिनेटली इट कंटेन्स डीएनए, peptidoglycan, typicolinic acid, keratin like coat. And mostly spores are present only in gram positive bacteria. This thing you need to remember. और इस spores को अपने survival के लिए resistance चाहिए होती है. Some level of resistance is required. तो ये जो spores होते हैं they are resistant to dehydration, heat and chemicals. किसी भी किस्म के chemical का, बहुत ज़्यादा heat exposure का या किसी भी किस्म की पानी की कमी का इन पर कोई असर नहीं होता. ठीक है. ये डोरमेंट रहते हैं और फिर जब फेवरेबल कंडीशंस आती हैं तो फिर से ये कॉलोनीज बनाने लग जाते हैं बैक्टीरियाज की. नाउ देयर आर अ लॉट ऑफ स्ट्रक्चर्स व्हिच आर इन्वॉल्व्ड in under the heading of cell envelope. The most important one is the capsule. You can see in this diagram, this is the capsule right over here, this one and this one. What does the capsule do? Capsule is organized, discrete polysaccharide layer except poly-D-glutamate on the P anthracis. You need to memorize this कि ये जो बैक्टीरिया है specifically P anthracis, इसके अंदर कैप्सूल प्रेजेंट नहीं होता. And what does the capsule do? It protects against phagocytosis. Then we have the glycocalyx. What is glycocalyx? It is a loose network of polysaccharides, and it mediates adherence to surfaces, especially foreign surfaces like indwelling catheters. जब भी operations या surgeries के दौरान catheters introduce कराए जाते हैं तो उनकी surfaces पर भी bacteria present हो सकते हैं. सही है. तो इसके अलावा we have the outer membrane. It's present in gram-negative bacteria only. You can see right over here. This is the outer membrane. और ये you can see right over here कि ये this is the outer membrane and it is composed of endotoxins and porins, whereas the outer membrane is absent in gram-positive bacteria. Outer membrane का outer leaflet है, inner leaflet है. The outer leaflet contains endotoxin. It may be lipopolysaccharide or lipooligosaccharide, and the embedded proteins, which include porin and other outer membrane proteins. So you can see right over here, this is the porin, these are the endotoxins. ठीक है. इनर लीफलेट के अंदर फॉस्फोलिपिड्स प्रेजेंट होते हैं. सो दिस इज द इनर लीफलेट और इसके अंदर आप देख सकते हैं फॉस्फोलिपिड्स. इसके अलावा इन्होंने बताया कि आउटर मेम्ब्रेन इज प्रेजेंट इन ग्राम नेगेटिव ओनली. वी हैव डिस्कस्ड इट ऑलरेडी. एंडोटॉक्सिन के अंदर जो लिपिड ए कंपोनेंट है, इट इंड्यूसेस द प्रो इन्फ्लेमेटरी साइटोकाइंस इंक्लूडिंग द ट्यूमर नेक्रोसिस फैक्टर एंड इंटरल्यूकिन 1. और जो पॉलीसेकेराइड कंपोनेंट है एंडोटॉक्सिन का, एक तो लिपिड कंपोनेंट हमने डिस्कस कर लिया जो कि ये प्रोड्यूस करे ट्यूमर नेक्रोसिस फैक्टर और इंटरल्यूकिन 1. जबकि जो पॉलीसेकेराइड कंपोनेंट है एंडोटॉक्सिन का, इट इज़ एंटीजेनिक. ये इम्यून रिस्पॉन्सेस को मीडिएट करवाता है. इसी तरह इन्होंने कहा कि मोस्ट आउटर मेम्ब्रेन प्रोटीन्स दे आर एंटीजेनिक. और जो porins होते हैं, they are involved in transport of materials across the cell membrane. You see these are the channels and they mediate the transport across the cell membrane. Now coming on to periplasm. Periplasm, it is the space between cytoplasmic membrane and outer membrane in gram-negative bacteria only. The peptidoglycan is present in the middle. So you can see right over here, this space, this is the periplasmic space, this space. It's present between the cytoplasmic membrane. You can see right over here it's mentioned, and the outer membrane. So this space is absent in gram-positive bacteria. It's present only in gram-negative, right? What does the periplasm contain? It accumulates components exiting the gram-negative cells, including hydrolytic enzymes, example beta-lactamases. So ये जो enzymes हैं, ये इनके इसके अंदर accumulate हो जाते हैं. These are the hydrolytic enzymes. Now cell wall is composed of peptidoglycan, for sure we know that. And what is peptidoglycan? It is a sugar backbone with peptide side chains cross-linked by transpeptidase. अब ये जो एक net like structure है cell wall, it's very rigid, it provides support and it protects against the osmotic pressure damage. तो बैक्टीरिया को जो भी osmotic pressure face करना पड़ेगा, उसको कौन protect करेगा? Cell membrane ही protect करेगी, slime layer नहीं protect करेगी, उसको cell wall protect करेगी. Now let's come to the last structure. It's the cytoplasmic membrane. It's evident right over here. It's present both in gram-positive and gram-negative. It's a phospholipid bilayer. You can see this is a bilayered structure with embedded proteins. Example, penicillin binding proteins, PBPs, and other enzymes. Lipotecoic acid, it is present in gram-positive only. It's mentioned over here. It extends from the membrane to the exterior of the cell, right? It's quite evident over here in the diagram. Now, cytoplasmic membrane is an important structure. It is a site of, as it is a site of oxidative and transport enzymes. The penicillin binding proteins, abbreviated as PBPs, they are involved in cell wall synthesis, so you need to memorize it. Function क्या होता है penicillin binding proteins का? इन्होंने cell wall synthesis करनी है. Lipotecoic acids, they induce the pro-inflammatory cytokines like tumor necrosis factor alpha and interleukin 1, right? So this is it for today. What we have discussed today is that the bacterial structures in basic bacteriology. in which we have discussed about appendages, which include flagellum and pilus. Flagellum is involved in motility, pilus is involved in adherence and conjugation. The spores are dormant structures which resist dehydration, heat, and chemicals. And cell envelope includes capsule, glycocalyx, outer membrane, periplasm, cell wall, and cytoplasmic membrane. And just go through this diagram, and you'll very easily understand the bacterial structure. So thank you. That's it for today. We'll meet in the next video, inshallah.